In the video tracking assay, flies must be loaded into a square observation chamber without the use of anesthesia. To do this, a small funnel must be lined up with screw holes in the chamber so as to knock the flies into the chamber using this funnel. Screws then seal the holes of the chamber, and an air pulse is administered through the air vents to initiate locomotion. A video camera suspended above the chamber captures this locomotion and sends it to a computer, where the paths of the flies are traced and analyzed.
Hi, I'm Justin Slawson from the Griffith Lab here at the Biology Department at Brandeis University. Today, we're going to show you a procedure for high-resolution video tracking of locomotion. We're going to use this procedure in our lab to study locomotor behavior in adult flies. So let's get started.
To get started with the experiment, grow flies in bottles containing yeast-free standard media. This protocol requires a large number of flies, so crosses need to be set up accordingly. Flies should be grown in a 12-hour light-dark cycle at 25 degrees Celsius. Collect the flies 1 to 3 days after eclosion. Use a carbon dioxide diffuser to sort flies into test tubes containing yeast-free media. Sort 10 males per test tube. Wait at least a day or two after use of carbon dioxide before running the behavioral experiments. Once the flies have been collected and sorted, they can be run in the video tracking system. The video tracking experiments are done in an environment room, which maintains a constant